I'm Dr. Michael Pickart. I'm in the biology department at the University of Wisconsin-Stout. And my office right now is in 184 Michaels, cubicle 29. In my lab, we have a few projects that we're focused on. The first one is a chemical genetics project where we are looking at using small molecule libraries to identify developmental phenotypes in zebrafish. We also have another project where we're investigating cancer stem cells with Marshfield Clinic and UW Falls. And so I guess the third project that we have that we're really involved in is we're actually doing genetic techniques for mapping mutations within the genome. The face for research in the future at Stout, I think, is just tremendous. We are increasingly going to allow students to experience science and technology. And generally the scope of the work, we have people working in many different areas, from environmental to developmental biology, like the things that I do. I think the scope is very broad for the kinds of research that we'll be able to do on this campus, and it will be very focused on emerging technologies and cutting-edge kinds of technology that you would hope to be exposed to as a student. Laboratories in our science building are of great benefit to the students in our program. I think mainly for two reasons. First of all, you do work in the laboratory on projects, and those projects are associated with faculty mentors and other students. In my case, we have student research teams, 4 to 6 students working on a particular project with me. And so you learn about working in a team environment and asking questions, research questions, and then actually doing that in the lab. We have equipment and other resources that are what's used in industry and used in graduate programs and used in academic research settings and all those places. You're not learning from watered down methods and watered down techniques. You're learning with the real stuff. Generally the labs are open from about 7:30 or 8 a.m. in the morning until about 9 o'clock at night. Generally, we just have to ensure that there are people around and that we're following safety procedures to ensure that everyone is working in a safe environment. But students can gain access to initially first the classroom that they were working in, where they have the materials they need to do the work. And then as you get connected more and more with faculty research on the campus, in my own case, then you would also gain access to GTAC and the genomics equipment and the zebrafish facility. And basically that access is on a case-by-case basis, where if you have a project, you'll be working with me or another faculty, and we'd figure out what your schedule is and when you would need to get in, and then we can control all the access that you would need through the card swipe process. So we have a lot of different labs, really, in the areas that I focus in. So let me talk about, there's 3 of them. The first one would be, we have the zebrafish facility in our animal facility in the basement of Jarvis. We have our Genomics Technology Access Center, GTAC. The other facility that I'm intimately involved with is the biotech classroom.